Botany and Black and Noah Hill. I'm gonna talk about some 101 shit today, and that's photosynthesis. Um, it's one of them things where you think you understand it, but when you really delve into it, you find out you don't know shit. Yeah, that's photosynthesis. I know we understand the concept and the general idea of it, but let's go into it just a little bit, and we'll find out how much we don't know. Alright? Let's start with what we do know. Pigments make photosynthesis happen. Alright? And we'll find these pigments within the chloroplast of plant cells, and the chloroplast is just a little plant organelle. But this chloroplast ain't some little sack of it's not this sack of chlorophyll. Within the chloroplast you got these complexes or these structures, and they're flat little membrane sacs called thylakoids, and one they're stacked up on top of each other, and that's called grana. And this is where your light reactions take place. We'll talk about light reactions and dark reactions, because photosynthesis, you see, it's a blanket term describing two main processes, your light and your dark reactions. So within the thylakoid membrane you have these proteins that make up your photosystem, and that's where you'll find your chlorophyll. If you look at a chlorophyll, the atomic structure of it, you'll see that it has it like a hydrocarbon tail, a lipid tail, and that's what anchors it into the thylakoid membrane. You see, or into the proteins. But if plants only use chlorophyll to do photosynthesis, they wouldn't be as efficient as they are, because chlorophyll only absorbs red and blue light. It reflects green, which is why plants appear to be green. And if that's the only wavelength to the EM spectrum plants can pick up, they wouldn't be as good as doing photosynthesis as they are. So you have other pigments like chlorophyll B, xanthan, fucoxanthin, and the famous carotenoids. Okay, these other pigments within that, as also in that protein, act as an antenna complex, because what they do is they're able to absorb these other wavelengths of the EM spectrum that chlorophyll couldn't absorb, and it focuses that energy to chlorophyll, which becomes your reaction site. Alright, so once that light reaches chlorophyll, that light excites electrons in chlorophyll until it moves to an outer energy level, a shell, until it eventually leaves that chlorophyll molecule and jumps to another protein. But while this is going down, you got some shit called photolysis going down. When that same energy from the sun via light, that splits H2O molecules, and what happens is it does that to replace the electron lost from chlorophyll, and it takes that electron from hydrogen and leaves us with the hydrogen ion. And nature isn't wasteful. We'll see that ion again, the hydrogen ion again. But we also have some oxygen, O2, in a gaseous state, which exits the plant, in which we breathe in. Just to keep it real with you, only like 20% of the Earth's oxygen comes from land plants. The large majority of it comes from blue-green algae in the sea doing photolysis and splitting them water molecules. So our oxygen comes from water, not CO2. Alright. So bam, we got some photolysis going down. Bam, we got electrons leaving chlorophyll. Bam, jumping to another protein. But the protein it jumps to is a protein and something called an electron transport chain. And that is exactly what it sounds like. It's a chain of proteins that transports electrons. And it's like each electron is more electronegative than the last. Okay. Got 3 kids. This one has a piece of candy. But this one is bigger and stronger, so he steals the piece of candy from him. But this one is bigger and stronger, so he takes the piece of candy from him. What I'm showing you is what I'm trying to explain is if these were proteins in the electron transport chain, each protein would be more electronegative than the last. Alright. But while this is going down, what you have is electron flow. You got the electrons going through the chain. And what one of these proteins in the chain does is uses the energy through the movement of electrons to pump those hydrogen ions for photolysis across the thylakoid membrane. Again, to be used for something else. Bam. So that electron is going through the electron transport chain and reaches this final electron carrier, NADP, which is reduced to NADPH. Because remember, when something accepts electron, it's reduced. When something loses electrons, it's oxidized. Okay? Back to that hydrogen ion. We pumped it across the thylakoid membrane, only for it to come back across through a protein called ATP synthase. Okay? And ATP synthase uses the movement of hydrogen ions to catalyze the formation of ATP by bringing ADP together with phosphorus to create ATP, or adenosine diphosphate, into adenosine triphosphate, 2 into 3. So far we made some ATP and we made some NADPH. But that by itself don't really mean shit, because plants need plants need carbon. It's composed of carbon, and it creates its food from carbon. And it doesn't get its carbon from the soil. You see? Plants get their carbon through openings in their leaves called stomata. Okay? But before we go there, there's something I kind of went past. We made some phosphorus, right? We used we pumped the thylakoids across the membrane, and then it came back through. That's called chemiosmosis. But we also have something called cyclic and non-cyclic photophosphorylation. And I'm going to just keep it short and simple. Cyclic is when that electron we use in chlorophyll is returned to chlorophyll. Non-cyclic is when that electron taken from water, it goes on through the works and it doesn't return. Okay, bam! I just had to put that out there real quick. I knew I was missing something. So, plants, CO2. How do they get it? Because it's not the soil. They get it from CO2 in their environment. Okay, that's how they get their carbon from the environment. These openings called stomata. Okay, CO2 enters there and immediately is attached to rubisco, an enzyme. And this enzyme takes this inorganic carbon and attaches it to a 5-carbon sugar called ribulose bisphosphate, or RUBP, which creates a 6-carbon molecule, which is then immediately split into something we call, into 2 molecules called PGA. Okay, 3-phosphate 3 phosphoglycerate, I'm sorry. But at this point, these two molecules are going to need help in their evolution to become whatever they to become next. And what to be used? Energy from ATP and electrons from NADPH. Okay, and then electrons from NADPH is going to reduce this molecule to a sugar called G3P, or glyceraldehyde 3-phosphate. And what a plant can do is use 2 of these little fuckers to make 1 glucose, and then that glucose can come together with another glucose to make your next sugar. Or you can add a little bit more of this or a little bit more of that and create ribulose bisphosphate to restart the process. Okay, and this is that's called, what we just explained is carbon fixation, and that's the first step in the dark reactions with the Calvin cycle, Calvin-Benson cycle. Because we call that the dark reaction because we don't necessarily need light to do carbon fixation, but we do require the ATP and NADPH made during the light reactions, okay, to make our sugars, to make our plant food. Okay, so plants make their own food, they're autotrophs. So them things you get at the store and stick in the ground, that's not plant food. A better term for it is fertilizer. Because with those things, they're adding minerals that act as cofactors to help out enzymes. So shit like carbon fixation can happen and other things. Okay. So I mean, it sounds like hella technical, but it's really not that deep. It's not. Alright, so bam.